Yeah. Hello, everyone. Welcome to the next lecture of MACSMIN 2023. It's a pleasure to introduce Professor Simon Billinge from Columbia University, who will talk about the following interesting question. Do materials have a genome? And if they do, what can we do with it? Please, Simon, over to you.
Yeah, thanks so much, Vitaliy. So I'm really excited about this meeting. I've been thinking about nanomaterials. I'm really excited about next talk. I'm really excited to introduce graph theory and the idea of patterns versus order and being constrained by crystallography, because we've also been thinking about these things. We're actually doing things more at the structural level, but I'm going to actually look at this idea about materials genome, which I'll explain to you as we go along. So first of all, yeah, I'm at Columbia University. Yeah,
You have to click the mouse on green.
okay, yeah. So this is actually New York City, and Columbia University is right here. There's Hudson River, Riverside Park is green, Central Park, which is green, and then Columbia University has some lawns.
Oh.
So some of the only green lawns in New York City. So if you come to New York City, you can come visit me in Columbia. And this is a yellow circle, and this is my plan for an X-ray synchrotron in New York City. So I submitted this plan to the mayor. I'm waiting to hear back. But until I get a positive determination, actually, we do a lot of structural work on nanomaterials, and we go and we do it at Brookhaven National Lab, which is about 60 miles down the road on Long Island. So they have a beautiful third-generation synchrotron source there. Okay. All right, so materials genomics. Well, okay, I have to take a step back. I'm not a biologist. I have no idea what a genome is. I know what a protein is, nothing. But starting in 1990, it seemed that the NIH and the DOE launched something called the Human Genome Project. So we can all remember that. And the basic idea was, okay, we'll sequence the human genome and and all good things will come from it. And so they did that, and it was remarkably successful. It ended ahead of schedule, under budget. It's like the most amazing project. And the basic idea is that once you sequence the genome, then you can try and do genomics. So you can make predictions just based on this genetic code. There's obviously a lot of biology in between the genetic code and the human being, but we can at least make some predictions just based on this code that will actually accelerate that journey. So yeah, again, I'm not a biologist, so I have no idea what a gene is, so I go and look at Wikipedia, of course we do. So it's a double helix DNA strand or something, and this seems to be like attached to a bunny. I'm not sure, but I think that thing there might be the ribosome or something like that. I don't know. It's too many biologists in the room for me to make progress here. But it's basically this kind of one-dimensional thing, but it's basically just storing a code. So another way of thinking about a gene, as well as a biological molecule, it's just kind of a set of letters split up into sets of 3, which are codons. And you need 3 of them because then you have to code for 26 proteins, and you use binomial mathematics to figure out that you need at least 3 of these base pairs to code for 26 proteins. And then once you have 26 proteins, you make sequences of codons, and that tells you the sequence of the amino acids, and then you get the proteins. So again, it's now a discrete code. It's not even discrete. It's not even continuous. So what I want to leave you with is a gene. If we define a gene as a 1D sequence of symbols that codes for a 3D uniquely folded protein structure, then that's what I want to call a gene. Okay. And if you have the gene, what can you do with it? Well, you can put it into databases. Once it's in a database, you can do all kinds of things on the database. So this was actually very recent, August the 12th, 2022, and this was a New York Times article. And hospital and drug maker moved to build a vast database of New Yorkers' DNA. So every New Yorker is going to have their DNA in a database, according to this. And then what people can do in principle is then again, use that large database to do data analytics, machine learning, things like that. Okay, what's the materials genome? So materials don't have a genome, but somehow, somewhere along the line in the US, what we're always trying to do is we're trying to persuade the government to give us money, and we try and explain things very simply. And the basic idea is there's always exciting applications of genomics that are revolutionizing healthcare. Why don't we have a genomics that revolutionizes materials science? And so we persuaded President Obama in 2011. He said, oh yeah, let's have the Materials Genome Initiative. So what is it? And basically what it is what they figure out is that the time and the cost of from a materials discovery event to actually putting it into practice in a device in operation takes something like 25 years and billions of dollars. And so if we take a genomic approach, in other words, a kind of a data analytic approach, that we can do better predictions and we can actually accelerate all of these things. So basically the idea was halve the time and the cost of materials discovery and bringing new materials into operations. And then we're going to do, so it's kind of a little bit hard to see, but behind here, there's accelerating our pace, and here's time to market. And then this is before MGI, like this, and then after MGI, it's going to go like that. Okay. And this is another way of plotting it. So you've got a time axis and a number of new materials to market, and this is old way of doing it, and this is a new way of doing it. So this is what materials genomics is. It's actually got nothing to do with materials having a genome. And so this it's actually a kind of a decade after that thing started, and so we look back and we say, was it a success or not? So there's papers that were published about how big of a success it was, and for me, it's a kind of a modest success. I'm not sure any of these are like totally revolutionized anything, but you know, there's been some progress, and so this is another thing. There's a materials project database now, so you can go to this database, and it has 150,000 materials and 200,000 users and things like this. But basically, this is just doing materials prediction using theory. So what they do is they run density functional theory on big computers at the DOE, and they generate new materials. They extract some kind of materials properties. But this is the basic idea. The idea was, let's use genomic methods, which are something to do with computation, something to do with databases, something to do with predictive stuff, and it's happened, but we haven't really reaped the benefits of it quite yet, but hopefully we can persuade them for another at least 10 years of funding it to get there. So in 2021, they renewed the MGI for another 10 years, and now we have a new logo. So this logo, somebody called it like a friendship bracelet, I'm not sure. So before it was, this was the old logo. So now we have a new logo. So we're off the race. Yeah, another 10 years. Okay. But coming back to this idea is the only problem that materials don't have a genome. So what's it all about? And then I started to think, maybe we should think differently. Maybe we should think, actually, can we think about what a materials genome is? Right? Does it have a genome? And remember, I'm using this definition. A gene is a 1D sequence of symbols that codes for a 3D nucleotide protein structure. And so I want to make a case that there's actually an experimentally accessible thing which looks like a genome, and it's this thing that I've been working on for many years, atomic paired function. So I'll tell you what that thing is. But once you get to that point, and you'll see, we'll take a step back and we can generalize a lot of the things that they're talking about, the stuff that Taylor's doing, stuff that Nick is doing, that actually these graphs can actually code for 3D structure in a kind of rather meaningful way. What's the PDF? So if you have a nanomaterial, if you have a crystal, you put your crystal on the machine, you rotate it, and you get a 3D structure, and then you can invert it, solve the phase problem, get where all the atoms are. If you have a short-range ordered material or disordered material, whatever you want to call it, it produces scattering that's isotropic. It's the same in every direction, and you can't do that kind of very fancy job So what you do is you just take your piece of material and you put it actually here, and you open the shutter, and then the X-rays come out, they hit your sample, and then they hit the detector. And you get these circular patterns. These are powder diffraction patterns, they're called. And when you have a crystalline material, the lines are very sharp. And as the size of the particles get smaller and smaller, the peaks get broader and broader in reciprocal space, until you have something that's very short range order, it's very diffuse in reciprocal space. And what we can do is we can do a bunch of fancy corrections that includes Fourier transform. I'm disappointed that you didn't talk about the Fourier transform, but this is the kernel of the Fourier.
Another hour.
This is the kernel of the Fourier transform. This is the Fourier transform. It's a sine Fourier transform, and you get this thing called the radial pair distribution function, which has a probability on this axis and a distance on this axis. And it's the probability of finding two atoms separated by that distance. So this is some kind of very short range order material. You see it's very flat out here. So this means that our material has kind of asymptoted to a uniform density at high R. So what we're always doing is we're sitting on an atom and we're looking at our neighbors, and far out in the distance, we just see the average density. But as things get closer, we see kind of hills and valleys and greater and lower probabilities of finding neighbors. And so you see very sharp peaks down here, which are very well defined local order. But because it's not periodic and perfectly in registry, as you go further out, there are rotational, orientational disorder and B factors and things, and finally it ends up with the average density. So this is the pair distribution function. It's experimentally accessible, and we can also calculate structural models from it, so we know how to compute the pair distribution function directly from a structural model, so we can do regression. All right, so what kind of things do people actually use it for? So I just picked kind of one example, and again, you want to motivate it a little bit in terms of it's useful for something. So one thing that you want to do is convert sunlight into solar fuels. So you want to shine sunlight on water, have water turn into hydrogen and oxygen, harvest the hydrogen, and then run a bus on it. And the way that you do that is you shine sunlight on water, you do all that. But somewhere in the water, you have to have an electrode, it's an electrochemical cell. And somewhere on the electrochemical cell, you have to have catalysts, because electrochemical cells, like trees, they have to actually convert the sunlight into chemical bonds using enzymes, what we call enzymes in biology. In organic materials, we call them catalysts. And it turns out that the very good kind of catalyst for this, what they call the oxygen evolution reaction, are these little tiny platelets of cobalt oxide, and they're floating around in liquid. And so we want an experimental measurement where we take this thing floating around in liquid and we put in X-rays and then get out the structure of these things, so we can tell the difference between what the internal structure of this object is, what the structure is at the edges. It turns out a lot of the catalysis takes place at these edges, whether they're these small little disks or whether they're larger disks, whether they're stacked or not. And these are all different measures of kind of hierarchical order, which Nick was talking about. You can also take this thing, which is starting to look like a protein now. So this is a cobalt oxide cubane. So you arrange the cobalt and the oxygen on the corners of the cube, and it turns out that's highly reactive, but quite unstable. So the idea here is, again, not our work, it's a collaboration with Don Tilley at Berkeley. And basically what they said is, let's take this little cobalt oxide. It's now a cluster of 4 atoms. It's starting to look like a metal center. We're getting closer and closer to biological enzymes. And what you do is you stabilize it by putting these organic ligands on, and actually you make this kind of coordination chemistry network with big pores. And then water can get in, or things can get in here, and then they can attach to that site, and then they can do their catalysis, and then they can detach. So this is kind of, inorganic protein kind of looking thing. But with our PDF, we can actually, again, do the PDF measurements on this, even if this thing is not long, doesn't have long range crystallographic order, and we can see whether this cobalt oxide cubane is present, because we get these signals in our PDF. So this is my day job, like, doing kind of real things. And then, you know, sometimes when I can lock myself in a room with no messages, I can think about other things. So the sort of stuff I'll talk about after this is about that. To get a better feeling for the PDF, we can use this beautiful C60 molecule. So that it's made of carbon. So carbon-carbon bond is 1.7 angstroms. And there's our first peak, 1.4 angstroms. So our first peak is there at 1.4. And then there's a second neighbor distance and a third neighbor distance on this hexagon. So that's the second neighbor, 2.54, 2.8 for the third neighbor distance. Okay, and you can see that actually the height of the third neighbor peak is much less than the height of the second neighbor peak. And that's because as well as hexagons here, we have pentagons. And pentagons have the near neighbor and they have the second neighbor, but they don't have the third neighbor. So the third neighbor is underrepresented. So this third peak is lower than it should be. So we can actually extract ring statistics from our sp2 graphite simply by measuring the relative height of this third peak to the second peak and so on. But we have this whole beautiful signal here, this is experimental data, by the way. We have this beautiful signal here. And if you look on this molecule, the last interatomic distance on the molecule, the longest one, is 7.1 angstroms, which is the diameter of the ball. And the last sharp peak is at 7.1. That's a 6 and that's an 8. I don't know what happened. So the red line is 7.1. So this part here is the intramolecular correlation. It's telling us the arrangement of the atoms on the molecule. And then what's happening out here? Well, what's happening out here is I have my molecules sort of ordered in space somehow. And I'm sitting on an atom on this guy, and I'm looking at an atom on that guy. And these things are spinning, they're dynamically reorienting at high speed because the data are collected at room temperature. So I can model that as having a sphere of a hollow sphere of uniform density. And I can make an FCC arrangement of these hollow spheres of uniform density. It turns out I get a triangular line shape for a hollow sphere of uniform density. And then if I look at this sphere distance, it's peaked at around 10. And it's FCC, so second neighbor distance is root 2 times 10, third neighbor distance is root 3 times 10. So I expect a second neighbor distance of 14, third neighbor distance of 17. And so this thing out here is actually telling us about the packing of the molecules. So I have a beautiful hierarchical function that captures my absolute local geometry and my intermediate geometry, and I see how it propagates out all the way to long range. And if it's a crystallographic and it's perfectly ordered over long range, then I see these sharp peaks extending to infinity. Okay, so let me make a proposition then. PDF is a genome for materials. Why not? Like, we've got us funding for 20 years. Let's keep some funding coming with PDF, right? The PDF is a genome for materials. So what I showed you is a measured PDF, which is a continuous function. It's continuous for a couple of reasons. One reason is the ADPs, the B factors we talked about, which broadens these intraatomic distances. Another is that we have finite measurement resolution, which broadens these, things like that. But underneath, if you look at this C60, you know, we can actually specify it as the atoms being centered at some location, and then we have a discrete interatomic distances. And what we do is we build a distance list, so that's exactly your distance matrix. But what we've done is we've taken the distance matrix where we knew what was on each end of every node and edge, and we've just compressed it down into a 1D function where we've thrown away the assignment information, but we've just kept the length. So now what we have is just this 1D list, and this is what the PDF looks like for this guy, which we would then broaden with our resolution and effects and stuff like this. But this is a 1D function of like discrete values that codes for a 3D structure. So this is our genome right here. If you do a generic graph, which is just, this is going from geometry in real space to graphs. Graphs don't have a geometry, they exist in graph land and they have a topology, right? And so this is a generic graph, which is actually one that basically is a random set of points. And then we don't have any that have the same length as each other, but we have the position of each of them. So this is much more of the graph theory end, this one. But the top one is where we've taken something that has geometry, turned it into something that has topology, and then compressed the topology by throwing away assignment information. Obviously, you hate throwing away information, but we're trying to map it back onto our measurable quantity. So PDF is a 1D discrete list of quantities that codes the 3D structures of our genome. So I did a bunch of work on the slightly more mathematical side of this with Phil Duxbury, who was a colleague at Michigan State. And basically we said, okay, we can actually, there's a distance geometry problem, which is a problem in mathematics, and it's somehow related to embedding distances into Euclidean spaces. And so there's two kinds of distance geometry problem. The one that existed before was actually the assigned distance geometry problem, where you have this distance matrix, and that gives you an assignment. So that gives you a graph which has nodes and edges, and you have the correct assignment. And the next step in the distance geometry problem is the embedding part of it, where you want to take your graph and you want to embed it into a Euclidean space. So you can actually express it somewhat mathematically. That's up here. But basically, you have sets of doubles, which are the nodes on your graph, and with them they have associated an L, a distance, and then the embedding is the satisfiability problem that these two sets are self-satisfy. It turns out that the assigned problem is quite straightforward to solve, but what we're doing is we're throwing away the assignment, so we don't know what the rows and the columns are in the matrix, but we're just taking all these numbers and putting them into an ordered list. That's the so-called unassigned distance geometry problem. And that's the one that we were working on, just really kind of specifying it. And then it turns out that's a hard problem. There's no uniqueness theorem, so there's no, in fact, we have definite violations. So we know that the mapping from here to, well, here to the geometry is not unique, but here to here is not unique. Actually, no, from here to the geometry is not unique. What we believe is that from a practical perspective, when the number of nodes in your graph gets large, it becomes quite unique. It becomes very, very hard to find something that will embed more than one thing that will embed when the size of your graph is sufficiently complex. It doesn't make it so easy to solve it. So this is where we were 2016, and we haven't taken it much further. So Phil became a dean of his school at Michigan State, and I was kind of doing other things. I didn't have the right students, but I'm very excited to come back to this. And also we started playing around a little bit with topological data analysis to see if there was any kind of helpful thing there. And then I'm learning what Vitaliy's doing. I'm like, okay, now's the right time to kind of turn back and revisit this. So what I'm going to do now is I'm going to say, look, we have a mathematical, your mid plus plus. Okay, we're in the plus column because we have some proofs and theorems. But what I'd like to do is take it further and start mapping it onto some of these ideas of coming from topological data analysis. So this isn't useful for anything, but we have a mathematical description. But it's a nice way to think about it, because it turns out that this embedding step, which is taking the graph and putting it into Euclidean space, is an enormously highly constrained, it's like a really high constraint to put on things. So we can start actually measuring PDFs. So I could imagine having a machine learning model where you have a measured PDF, you extract a distance list, and then you just see whether it embeds or not. If it embeds, it's a structure. If it doesn't embed, it's a multi-phase pattern. Right. So there's lots of things we can do. Okay, so now I'm going to take a step back and I'm going to say, well, what were we doing before that? So we were trying to solve this problem of ab initio structure solution of nanoparticles from this 1D distance list. So we extract the 1D distances with some uncertainty. And then what we end up with is a list of distances. And so we wanted to come up with an algorithm that could do this reconstruction. We're not taking advantage of the UDGP here, we're just kind of doing it in space. And what we wanted to do was we wanted to say, okay, let's say that our target is this guy, the octahedron. what's the PDF of the octahedron? Well, it actually has distance 1 times 4, distance square root of 2 times 2. So it's the edges and the body diagonals. So this is our input. And we want to actually have an algorithm that will generate that. So what we do is we say, okay, our algorithm is the following. Pick an atom and pick a distance in our distance list, and then pick another atom. So the first thing is we lay down these two guys. And then we say, okay, let's take another atom out of our bag of atoms, and we want to place it on that cluster. We'll triangulate it. So we'll pick 2 atoms on our cluster, and then 2 distances from our distance list, and we'll triangulate onto that thing. Now, our first 3D object is when we take a 4th atom out of our box, and we want to triangulate it onto our 2D structure. So we now have to pick 3 atoms on the cluster, 3 distances from the distance list, and we triangulate it on modulo 2, because it could be on the left or the right side. What happens here is we've actually generated a wrong cluster, because this is our target, and there's no tetrahedron, which is a subcluster of an octahedron. So we need a backtracking method. So this is showing 0 error. The color of the balls is the error, and the blue means no error. So from our perspective, this is a fantastic cluster, but it's fantastically wrong, right? But we don't find out it's wrong until we build out the cluster. So we add 2 more atoms here, and then we start getting error. So we know we're up a bad path. We can actually quantify the error because we can actually compute the contribution. We know what our distance list is, and we can measure all the distances on our cluster, and we can see which distances on our cluster are not in the distance list, and they're contributing error. And this guy is worse than these 2 because there's a distance from this one to this one to 2 atoms. 2 atoms are contributing badly to that guy. So this guy here is bad. So what we'll do is the reason it's called Liga, it's based on soccer leagues. And so what we do is we have these clusters competing with each other. And then the best performing clusters, we promote them to a higher division. Higher division means more fans, bigger stadium, more money. So they buy players, right? then they're competing. So this one won the competition, had no error. It got promoted, it bought these 2 players, then it started performing very badly. So performing badly, relegate, go down to a lower division, have to sell some players. And of course, you sell your worst player, you're fairly smart, so you sell that guy, and then you end up with this. And then this one is performing well in the new competition, gets promoted, and then wins the premiership. So that was called Liga, it was fun, it worked. So we solved the nanostructure problem. We have it was really efficient. It's running on a laptop. We can solve really large clusters. So it seems like the job is done. You can see that we're using our prior information extremely efficiently in this algorithm. That's why it works so well. Because we're not using anything stochastic where we're randomly placing atoms, blah, 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 because we're selecting distances from the distances. So this is one example where having our genome is to our advantage. Okay. Fantastic. We solved all these crystal structures that have been solved crystallographically and were in the literature. So the algorithm is dynamite. So I should be very rich and famous at this point and not talking to you guys, but sitting on a yacht in Monaco or something, right?
Are you implying we're not rich and famous?
Well, everyone's famous. Yeah. So why is that true? Why am I not on my yacht in Monaco? And
Get bored.
and this is the problem. Like we solved C60, no problem. But we had this other cadmium selenide nanoparticle clusters that are atomically precise. We got them from very talented chemists, and that had 64 atoms. Successfully solved, failed to solve. The difference between them is this is what the data looked like for C60. That's extremely rigid molecule. And this is what it looked like for this 64 atom nanoparticle cluster. And you can see just quality of the information content in the data is much lower. So Pavel, who's my very talented postdoc who actually did the legal work, he said, okay, I'm going to go to the crystal structure that the CSD, the Cambridge Structural Database. This is molecules. And I'm going to, because I'm a mathematician on it, chemist, I'm going to say they're all made of carbon. So I'm just looking kind of at the geometry, whether it makes chemical sense or not. I think he may have got rid of the hydrogens, I'm not sure. So then he just like generates the distance list and sees whether Liga solves it. And so what he found is it sometimes solves it and sometimes doesn't. So this is the number of atoms in the cluster. So plus means success. No, star means success, plus means failure, right? And so look, we're having success with huge clusters, like 150 atom clusters, and we're having failure with tiny ones. But you can see that they separate on this graph, right? And so what's going on here? Well, what's this graph? This is the number of atoms on the cluster. This is the number of unique distances in the object. So if you have a large number of clusters, a large number of atoms, and a small number of unique distances, it means you have high symmetry, and vice versa. And so things that are up here near the top have low symmetry, and things that are down here have high symmetry. If you have a completely random graph, you have actually n into n minus 1 over 2. So there's this envelope that's completely random. You can't live up here. But anything can be below that line. And what we see is, again, if there's this line of death, basically, which if you're above this line, we can't solve you. If you're below this line, we can solve you. It's related to symmetry. So it's fundamentally an information theoretic problem that we're bumping up against, right? And the information theoretic statement is that we have a certain amount of information in our data. We have a certain number of degrees of freedom in our model. In principle, if the information content of the data is less than the degrees of freedom in our model, it's unsolvable. It's an ill-posed inverse problem. And this is what the data would look like for the low symmetry stuff. This is what the data would look like for the high symmetry stuff. You can just look, there's much more information in this guy than that guy. And so the problem for us, and the reason I'm not in Monaco on my yacht, is that most of the interesting problems actually fall above this line here. So, so this is the well-posed, this is the information content of the data on the left. This is the degrees of freedom in the model on the right. Well-posed, we can solve it in principle, and the ill-posed. And we have a great algorithm. So if we can solve it in principle, we can probably solve it in practice. But this is where we find ourselves, which is most of the interesting problems are these ill-posed ones. So now we have to start working on that aspect of it. So now we're just kind of trying different data analytic approaches. I'll just very quickly show you a few different things in case there's something of interest to you. But what I'm actually more interested in this forum with these people here is actually thinking about whether we can take this kind of genomic view, you know, TDA, graph theory, how far we can take it to actually try and, in some sense, regularize this ill-posed inverse problem. So this one here is trying to actually solve a problem that we encounter a lot, which is we have a flat PDF at high R, and then we have like 5 bumps at low R. And we know a little bit about the chemistry. So this is Kirsten Jensen, who's my former postdoc, but she's a faculty at University of Copenhagen now. So we'll measure a PDF that looks like the blue line. And actually, in this case, she knows what the actual structure is, because I think there was maybe a crystallographic solution or something, and it's this kind of alpha kegging cluster, which is W12040. And so we were using Liga, where we were just trying to start from this place and build up. But she had the idea of, instead of having a cluster build-up, why don't we have a cluster construction? Why don't we have a cluster deconstruction? So the idea was to go into the crystallographic database and say, if this is a low energy thing, it probably exists somewhere in the database. So if we can actually find a bunch of structures that have the right chemistry, and then what we can do is we can start deconstructing them to find the subcluster of the bigger cluster, which is the right one. And so this actually worked really well. This was actually a program that was developed by Andy Anker, who's a postdoc working with Kirsten. And he used actually explainable machine learning, which he used something like the SHAP measure to look at. You've got this cluster and you're decomposing the cluster, and then you're measuring the contribution to the cluster, the atoms that are contributing the most error, and you're getting that from the SHAP measure, and then doing it. And so it worked. And so I think the reason it worked is because, again, we're regularizing the problem by only sampling from structures that are somehow low energy because they already exist in a database. So we're bringing prior knowledge to the problem that Liga can do on its own. So it worked very well. So this is what the answer looks like. So basically what you do is you go into the structural database, you cut out a big chunk of material, and then you just run it through this machine learning algorithm, and it will show you which atoms it thinks should be in the cluster and which ones it shouldn't. So it does it with some error. It found this Kagan cluster in actually 1, 2, 3, 4 different crystal structures of greater and greater complexity. But this one kind of worked very well. So that's one approach. You take prior knowledge and then you deconstruct from it. I don't need to do that. Okay. Another thing that we wanted to do was actually, we've got our genome now, which is the PDF. And so the PDF is this 1D function. You saw what it looked like for C60. And then we have crystallographers, and crystallographers, they love their space groups, right? So the first thing, you give them a diffraction pattern, and they want to know what's the space group. So I could give you then a PDF, and I could say, it's a perfectly legitimate question, like, here's a PDF, what's the space group? There's no direct way of getting from the PDF to the space group, because there isn't. You could in principle Fourier transform it and do all that, but so but the information about the space group is in the PDF, because the PDF is just the Fourier transform of the reciprocal space information, which contains the space group information, and the Fourier transform is a linear transform which neither adds nor removes any information. So a priori we know that the information is in the PDF, but we just don't know how to decode it. So that's like a perfect machine learning problem, because you can say, okay, well, I don't understand how to do it, and the machine learning model doesn't understand how to do it. The machine learning model just can do it by spending hours and hours, you know, comparing things and updating its linkage weights. So the idea was look at a PDF and just, so input is PDF, output is the space group. And it worked. So we used, in the end, the thing that worked the best was the CNN, convolutional neural net. This is what the structure looks like. We had 100,000 structures out of the ICSD. We only did it with 45 space groups. It's obviously 230 space groups, but a lot of them are barely populated within structures. So this is the 45 most heavily populated space groups. We did a 80-20 training validation split, or training testing split. We tried logistic regression, but actually the CNN works really well. And this is actually plotting the classification ratio versus the space group order. So the number of a space group, it's the higher the number, the higher symmetry it has. So the better you would expect to do, and that indeed is the case. The size of these dots are how many examples of that thing was in the database. We did some normalization to more evenly weight them, but we wanted to show on the plot whether it does indeed. So logistic regression was all over the map actually, but here the CNN, it does better on things it can learn better, and does less well on things it can learn less well. But interestingly, we did the matrix of confusion. So this is the, on this axis is the actual label, and then on the top axis is the predicted one. And if your machine learning model is working perfectly, this should be diagonal matrix with ones on the diagonal. And down here, it's like, you know, working really well. But there are some actually misclassifications, and it turns out there's a certain small number of space groups that get preferentially misclassified into. So the machine learning model isn't just randomly getting things wrong. It's getting things wrong with a lot of underlying structure. And it turns out that there was actually physics in that these misclassifications are all subgroups of supergroups. And so we could work out the subgroup supergroup relationship by these teardrops of misclassification that were happening. And it actually turns out we went back and we looked at the literature in assigning space groups, and actually the space group algorithms that are deterministically done or not done with machine learning, they also have difficulty with these same things. So we actually, this is something I like very much, because we kind of learned some crystallography from the machine learning models. because I didn't actually know that. I know about space group subgroup relations, but I didn't know about this difficulty of disambiguating certain space groups. And what we're doing now is we're trying to solve, again, I'm still dreaming about my yacht in Monaco. So we're trying again, and this time we're just going for broke, and we're just going to do the whole thing in a machine learning model. So we're using a graph convolutional variational autoencoder. And basically what you do is you start with a structure, you put it through the encoder, and it maps it onto a point in this low dimensional latent space. So the encoder does structure to latent space mapping. And then you have a decoder, which does the point in the latent space back to a structure. So you start with this, you go through the latent space, and you end up with this, nothing happened. But you went through this low dimensional latent space. And so what we can do is we stuck to close packed like SEC, HCP, decahedral, icosahedral, close packed structures. And so we do this mapping in here, but every time that we map a structure onto a point in the latent space, we associate a PDF with it. So now we can actually, so I don't know, the machine learning people call this conditioning, but as far as I'm concerned, basically we're doing this mapping, this green thing to some point here, and then back to this green thing, but we're associating this. And so what we can then do after we've trained it is we can give a PDF, and it will know how to map the PDF to a point in the latent space. And then it can decode it to a structure. So that means that we can do generative, we can generate structures that it's never seen before, because we will be mapping to a point in the latent space that we don't have anything in from our training set, and it will decode it. And it actually worked brilliantly for this particular case. And actually also again shows you physics. So it turns out that it was a 2D latent space, and so this is a very nice thing to visualize. And so all of the HCP related structures are the pink ones, and decahedral ones are the orange ones, and SEC are these light blue ones. And there's some simple cubic ones, but the decahedral ones are like between HCP and FCC, because you can go from FCC to, this is for the crystallographers in the room, not the protein ones. You can go from FCC to HCP by introducing stacking faults. So FCC is ABC, ABC stacking, HCP is AB, AB stacking. So a stacking fault defect is ABC, ABC, AB. I've got a tiny little bit of HCP in my FCC. And then I can completely interpolate from FCC to HCP, which makes this very perfect, because what I'm going to do is I'm going to pick a point on my latent space, which interpolates from one that I know to another that I know, and it has at least a fighting chance to decode it correctly. So it worked brilliantly. And these decahedral ones are basically little chunks of FCC with these twin boundaries, and the twin boundaries turn out to be little tiny chunks of HCP. And so they should appear in between the HCP and the FCC. I know that's maybe too much information, but so, but in general, we want to do more with databases. So instead of having chemists, having crystallographers work with their laptop, and then the laptop dies and that's the end, or chemists doing synthesis and writing in a notebook. Ideally, we want to definitely push in the direction where this stuff is stored in databases, because we can then do much more kind of genomic and things. And I'm not going to go through this, but we're definitely coming up with ways of screening large numbers of models against data, where we're pulling these large numbers of molecules from databases as a general approach. And then we've got this website where we actually started putting things up on the web. And what we're doing is we want people to upload their PDF and then get some valuable things. For example, is our space group miner. So they give us the PDF, we give them the space group. We hold on to that PDF and we put it in the database. We tell them we're doing it. And then we're starting to build up a database. And in principle, we can start actually then doing things like extracting distance lists from all of the PDFs in the database. And then again, we're enriching the community's knowledge about what's going on. So that's just the general direction. And I couldn't resist to put this in. So this is machine readable scientific literature, which is actually collaboration with Nicola and people at ICR. And basically the problem is there are too many papers. We don't have time to read them. And so what we do is we spend time writing them, and then we publish them, and then we think, oh, I don't have time to read this, so I'll make a machine that can read this paper. That's called natural language processing, right? So we've written a paper that was written for a human to read, and then we've built a machine to read the paper that was written for the human to read. So my point is that we should build the paper for the machine to read in the first place. So we don't need to build a machine to read the paper that was written for the human to read. Yeah, so this is the machine readable project, and we're working with IUCR on this. Yeah, right. So I don't want to. I think we're running behind, so I'm not going to go through this, but I think you got the idea. I think these, genomics was kind of just a fun thing, but actually I think it's valid that to think in this way, to think in terms of graphs. I'm a very strong proponent of that, hierarchical structures. You can have a concept of distance-dependent symmetry, things like this. I mean, there's all kinds of things that can come out from it. I think it's very rich. I think it's going to grow. Happy to have that. We're all here to talk about it. And I tried to mention the students who are doing the work when it was done, but also we use the facilities and we get funding from DOE and so on. And then this is a group in times of homeless. What's it called? I can't remember. Yeah. Anyway.
They're not all together, sorry.
No, not by any means. So that's it. Yeah. Thank you.
But you still record them and you think about questions.